We're going to be on page 65, and we're starting to study human genetics and biotechnology. Remember, when we study human genetics, sometimes we're going to be looking at diseases, genetic diseases that you inherit. So these are problems with your DNA that cause a problem with a protein that give you a disease, such as cystic fibrosis, sickle cell anemia, Huntington's. Biotechnology is how can we solve problems. Technology is all about solving problems using our biology knowledge. So we're on page 65, and when we look at problems studying human genetics, so there's something that's called a test cross. And this is one of the things that we can do to help us study genetics, because remember, when we're studying genetics, we have this problem where we have to identify who's dominant and who is recessive. Remember, if you have a dominant phenotype, you could be either of these. So when we're studying genetics, sometimes we want to know if someone is a carrier. So we use something called a test cross. We take our unknown genotype and we mate them. So next to the word compare, if you'll write the word mate. We mate them with a homozygous recessive, because we know what genes a homozygous recessive has. So we mate them with that. And if any of their children turn out recessive, it means that they got a recessive from both parents. And so that's a test cross, where we purposefully mate you with someone who's a recessive to see if you're hiding any recessive alleles. But it's not really responsible to conduct test crosses on humans. Not many people want to sign up for a breeding experiment to mate with someone who has all recessive genes. Another problem is that humans have really long lifespans, so it can be decades between generations. And we usually have a small number of offspring, which means that even if we have the first one dominant, sometimes we have to have multiple offspring for any of the recessives to show up. So it's pretty hard to study human genetics. And so what we have to do is we have some specific techniques that we're going to use. The first is called population sampling. This is where we look at a small, randomly selected group. So maybe just Butler High School. Take a random group of Butler High School. Maybe we randomly pick 5 classes, and we count how many people, we sample, how many people can roll their tongues. And then we use that as a percentage. Well, if 10% of people at Butler can roll their tongue, then we would assume that maybe 10% of the general population can roll their tongue. But one of the most useful tools that we're going to use are pedigrees. These are records of inheritance of a single trait over several generations. We look at family history, we look at documents, interviews, but really what pedigrees can also help us do is determine the pattern of inheritance. What we mean by pattern of inheritance is it dominant? Is it recessive? Etc. So when we look at a pedigree, we might have a dad and a mom and their 3 kids. And we see that their son has a disease that the 2 parents don't. We know that the only way for it to be hidden is for it to be recessive. So that's what we're going to be doing, is looking at these patterns and determining whether having it is dominant or not. The shapes represent individuals, and the connecting lines represent relationships. Next to female, if you'll write curvy. Females are curvy. So we can remember that females get a circular shape. Males get squares. If you do not have it, unaffected means do not have it. But we're going to put it in quotation marks, because what is it? Is it cystic fibrosis? Is it hemophilia? And before we just memorized that cystic fibrosis was recessive, we had to track it over family generations. So affected means that you get filled in, and it means you do have it. But now we have to figure out, is it recessive or is it dominant? And so we have some rules to help us do that. The first question to consider, you always start by asking yourself, is having it dominant or is having it recessive? And basically what we mean is, does the filled in shape mean that it's dominant or recessive? So when we look at a pedigree, first we ask ourselves, is this a dominant or recessive? And the question that you ask is, are there any parents, any children who have the trait but both parents don't? If yes, then the trait is recessive. If no, then the parent passed the trait to the child, and then it's dominant. Usually. And so really what we're looking for is we're looking for shapes like this. A dad that doesn't have it, a mom that doesn't have it, and a child that does. Here's a child who has it, but both parents don't. That means it is recessive. So if you see two parents that don't have it and a child that does, ding, 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 it's recessive. And that helps us answer half of the questions. If it is recessive, then we want to know if it's autosomal or sex-linked. Autosomes are your chromosome pairs 1 through 22. Sex-linked are X on your X chromosome. So here's the question we want to ask. If a daughter has it, did her dad? And here's why. If a daughter has it and it's sex-linked, then the daughter would have to be X little b, X little b. So a lot of times we just guess, well, maybe it's sex-linked. If so, she had to get an X little b from her mom and an X little b from her dad. But her dad would have to be X little by, so he would have to have it. So if a daughter has it and her dad has it, then it is sex-linked. If a daughter has it and her dad doesn't, it's not sex-linked, it's autosomal. So that's the first thing that we ask. Then we might look at a mom who has it. If a mom has a sex-linked disease, she would be X little b, X little b. And this means that her sons get the X from her and the Y from the dad, which means every son has to have it. So if you ever see a mom who has it, doesn't matter what the dad has, if all of her sons have it, notice the squares are the boys, if they have sons and all of her sons have it, then it's probably sex-linked. If it is sex-linked, a son cannot inherit it from his dad. His dad might have it, but remember, his dad would be X little by, and he's giving the Y to the boys, not the X. So these are difficult questions. We start by asking if it's dominant or recessive, and then we try to figure out if it's sex-linked. It all has to do with, is it on those X's? If you answer no to both of these, then it's probably not sex-linked. So let's look at some examples. You are going to need to memorize these questions. So here are the questions again. Are there any children who have the trait but both parents don't? So if we look here, here we see two parents that do not have the trait and a child that does, which means that it is recessive. However, when we look at a daughter who has it, but her dad doesn't. Here's a daughter who has it, but her dad does not. So it cannot be sex-linked, and that's why it's autosomal recessive, because the daughter had it, but her dad didn't, so it can't be sex-linked. Here, we don't see any parents that don't have it. Both parents have it, a child has it. One parent has it, a child has it. If we look over here again, we can see two parents that don't have it and a son that does have it. So this tells us that it's recessive. Here, when we look at the mom who has it, her two sons, both of her sons, now this one isn't her son, this one married into the family. Her two sons have it. When we look at the daughter who has it, her dad has it. So that tells us that it is sex-linked. Now real quickly, we just want to talk about how we could possibly cure these diseases. One of the big things that we can do is look at something called the Human Genome Project. We can use pedigrees to track diseases, but now we can actually map the genes of humans, meaning that we actually go in and figure out the exact nucleotides that someone has. And we can look at gene variations and tell if someone has a disorder. So we now have the technology to actually determine what genes you have instead of just tracking if you have the phenotype. Because all that a pedigree shows us is the phenotype. The hope is that one day we could use gene therapy, which means we could fix your genes. Right now, that's not really a possibility because that gene is in every single one of your cells. And in order to fix those genes, we'd have to fix every single one of those cells. There are some preventions or treatments. The first is genetic counseling. This can help parents determine the chances of passing a harmful trait to their child. If someone has a genetic disease, we cannot cure it, but we can treat it. We can use pain medication to relieve the symptoms, like of sickle cell anemia. We can use occupational therapy, which is kind of like physical therapy. It helps improve their ability to perform tasks like speech. We can use blood transfusions, and we can think about using gene therapy. But we do have to be careful because right now gene therapy is not possible. It's just a hope. So someday we might be able to actually cure a genetic disease. This is the end of the section, so if you'll make sure that you fill out your summary box, and you probably want to go back and practice on those pedigrees.